Very good morning, my dear students. So today we'll be discussing one of the important cytogenetic concept, that is principles and applications of cytogenetics. So before going to the what is cytogenetics, so here this is the outline and agenda of this today's topic. So what is cytogenetics and what are the chromosomes and what is the chromosomal abnormality with respect to numerical and structural abnormality and applications of cytogenetics, and there are various samples used for the cytogenetics and cytogenetic analysis techniques. So before going to cytogenetics, so what is this cytogenetics? Cyto means cell, genetics means study of chromosomes. So here, so it is a branch of genetics that deals with structure and function of a cell throughout visualization of analysis of a chromosome. So if you see, this is in a pictorial representation, so where chromosomes are seen, so it is present in the inside the nucleus. So when it so this slide tells about, so this is the structure of chromosomes. So the chromosomes has a two arms. So one is P arm and Q arm. So P arm is nothing but short arm, Q arm nothing but long arm. So both these ends are joined together. You can call it as a centromere. So the ends of chromosomes are, you can call it as a telomere. So if you see, this is a 17th chromosome, it has a submetacentric. 21st chromosome as a acrocentric. So it has a satellite and stalk in the chromosomes. So this is a karyotype, you can call it as so arrangement of a chromosomes based on their size, length and binding patterns. So this is a normal human karyotype. Chromosomes are arranged. So this is a 22 autosomes and 2 sex chromosomes. So it may be XX or XY. So always chromosomes are present in a phase. So what is this chromosomal abnormalities? So chromosomal abnormality is nothing but, so there are different types of chromosomal abnormality. So based on their exchange of chromosomal segments. So under this, there is a translocations. So translocation is nothing but transfer of one chromosomal segment to the another. So under we have a deletion. So within a chromosome, a part of segment is deleted. So other chromosomal abnormality is called as inversion. So part of chromosome is inverted in the particular chromosome. So isochromosome. So one part of chromosome is joined together and there is a mutual sharing of a chromosomal segment. So other last chromosomal abnormality we can call it as a insertion. So a part of chromosome are inserted to the other chromosome, we can call it as a insertional chromosomal abnormalities. So what are the applications of cytogenetics? So we know that chromosomes are, so based on their size, binding patterns and centromere positions are vary in the first chromosomes to 46 chromosomes. So what are the applications of chromosomes? So usually, so cytogenetics deals with the chromosomes. So here, so the neoplastic diagnosis. So it is usually seen in the cancer biology. So and also there are several congenital abnormality diagnosis can also be seen. So under there are several prenatal diagnosis in terms of they are taking a amniotic fluid and cord blade from the mother womb, and also we need to check whether X or Y related to the syndromes per particular thing. So what are the various samples used for the cytogenetics? So the first sample is peripheral blood, and the second sample is fibroblast from the skin biopsy, and epithelial cells from the buccal smear, bone marrow, that is called hemoblastosis, and solid tumor biopsy also can be taken for the rule out the cytogenetics whether chromosomal abnormality is there or not. So what are the different techniques for the cytogenetic analysis? There are three types of techniques. So one is high resolution karyotype and fluorescent in situ hybridization, array based comparative genomic hybridization. So the most usefulness of our numerical chromosomal abnormality is, so usually we will use for the fluorescent in situ hybridization. So these are the, when compared to conventional cytogenetics, so these are chromosomal analysis can be carried out. So based on their structural or numerical abnormality. So before going to any structural abnormality, so we need to know what is the flow scheme of karyotype, means how we are getting from the cell to chromosomes. So first we need to take a blade, peripheral blade or tumor tissues, or we need to culture them using a RPMI media. It is called as a Roswell Park Memorial Institute is a media. So we need to culture, and along with a fetal bovine serum with an antibiotics. We can culture as a penicillin or streptomycin. So here, so after 72 hours of exact culture in a CO2 incubator at 37 degrees centigrade, so we need to harvest them. So before harvesting, so we need to stop the cell division at the exactly at the metaphase stage. So for that reason, so we are using a colchicine is a chemical to stop exactly at the metaphase stage. So after that, we need to treat the cells with a certain procedure. So we need to hypotonic treatment and fixative steps can be followed. So later after the harvesting, so we need to call the cells. So whether the chromosomes are there or not. So we need to prepare a test leads. So the chromosomal, whatever the harvested sample, it has a good chromosomal index. So after that, you need to stain with a Giemsa stain and followed by G banding. So G banding is a Giemsa stain, trypsin is an banding to band the chromosomal techniques. So based on there, so this is entire metaphase chromosome obtained. So I told you that high resolution banding. So high resolution karyotype are techniques. So where the bands of a particular chromosome can be seen in a resolution will be high when compared to the conventional cytogenetics. So what are the advantage of this high resolution karyotypes? The advantage is whole genome scan can be observed, relative low cost when compared to the fluorescent in situ hybridization. So what are disadvantage of this high resolution karyotype is? So labor intensive, highly dependent upon operator experience and skills can be needed. So detect major structural abnormality. So one band almost 6 MB of DNA. So it is nearly, so 150 genes can be detected. So what is this fluorescent in situ hybridization? So this we can call it as a FISH. So it's a process of where the chromosomes are finds and detect the RNA as well as DNA structures, numbers and location in place of a cell with help in the place of a cells with help of in situ. So this is a fluorescent microscopy. So where you can capture the whatever the cells, so using a fluorescence probes. Okay. So this is a flow scheme of how fluorescence hybridization can be taken place. So this is a double stranded DNA. So where the label with a fluorescent dye with this double stranded DNA. So wherever there is a chromosomal abnormality, it will go and bind and it will give the fluorescent signal. And this fluorescent signal can be captured through the fluorescent microscopy. So this is all about introduction of cytogenetic principles and applications of. Thank you very much.